Alright guys, the last video for this week. Whew, it's been a lot. So great job of keeping up for me this far. So far we've talked about what a cell is, all of the organelles inside of it, the role of the cell membrane, and how molecules in water move around the cell, inside and out. The last thing we need to talk about is how cells reproduce. To do that, we need to talk about DNA. So if you remember from the chemistry lesson, DNA is deoxyribonucleic acid. It's a nucleic acid that has a sugar, a phosphate, and one of 4 nitrogen bases, either adenine, guanine, thymine, or cytosine. So when we see DNA, we often see an image like this. A twisting helix with these bars in the middle, and these bars are called nucleotides, and they're pairs of those nitrogen bases that are bound together. In DNA, adenine will only bond with thymine, and guanine with cytosine. So when you start stacking these pairs in a particular pattern, you end up with a gene. A collection of genes is called a chromosome. So why do we care? Well, the purpose of DNA is to carry information, and that pattern is very particular. It's a certain order, and it tells the ribosomes outside of the cell what shape and function the proteins that they're making are going to have. These proteins will determine how our body looks and functions, everything from our eye color, hair color, muscle mass, height, everything. And as we age, they'll determine how our body handles things like disease and injury. It's really important stuff. Okay, so DNA has the information. It's called a gene. And all the DNA is inside the nucleus, but we need it to get outside of the nuclear envelope to help the ribosomes to make proteins for other molecules. The problem is, DNA is really big, and it's too big to fit through the nuclear pores. So what do? We use RNA, specifically messenger RNA, mRNA. The DNA unwinds and separates, and it's read by a piece of RNA. The RNA matches with particular bases, just like in DNA, so adenine goes with uracil, and guanine still goes with cytosine. Once the RNA has the pattern down, once it's figured out what the DNA is trying to say, it leaves the cell and meets up with a ribosome, who takes that information and converts it to a protein. The reading of DNA by mRNA is called transcription, and the decoding of that RNA by a ribosome is called translation. It's kind of like that secret code stuff that you see in spy movies. Someone writes a message, puts it into a code, and then the recipient needs to decode it in order to get the message out. It's pretty cool. Alright, last topic. So we have this cell. It spent its whole life making proteins, carbs, lipids, and it's done a really good job. But now it's ready to reproduce and make new little baby cells. So for most cells, that means mitosis. Essentially a cell splitting apart and making 2 carbon copies of the original cell. Each new cell will have the exact same genetic sequence or number of chromosomes as the original one. For humans, that means each cell has 46 chromosomes. Certain cells instead will go through meiosis, which means the end product only has 23 chromosomes. These cells in particular would be sperm cells or egg cells, and this allows us to pass on our genetic information to our offspring while also inheriting the traits from our partner or for the other parent. So mitosis or cell division has 4 phases. In the prophase, the loosely coiled DNA begin to tighten up, forming chromosomes and beginning the process of duplication, creating chromatids. Centrioles, the organelles that organize and distribute other structures in the cell, begin to move to opposite ends of the cell, and at the end of this phase, the nuclear envelope dissolves. In the metaphase, spindle fibers attach the centrioles, which are out to the sides of the cell, to one of the pairs of chromosomes, from one side or the other. And then they begin to line all the chromosomes up in the middle. In the anaphase, the centrioles pull on the chromatids, splitting them apart and back into their chromosomes, and then pulling each one of the sides to the cell, or to the new cell. Finally, in the telophase, a new cell membrane and nuclear envelope is formed. The nuclear envelope forms around the chromosomes. The cell membranes pull completely apart, making brand new cells.